Assalamu alaikum wa rahmatullah. In this video we are going to keep talking about the reaction of cellular respiration. So in the previous video we already saw that cellular respiration can be broken down into three main phases. The first one is the glycolysis, the second one is the Krebs cycle, and the third phase is the oxidative phosphorylation. So in this video especially we are going to talk about the second phase, which is the Krebs cycle. Well, before getting into the Krebs cycle reaction, we already gone over the glycolysis pathway, in which we converted one molecule of glucose into two molecules of pyruvate. And during this process we generated 2 ATP and 2 NADH+. These reactions happen in the presence or the absence of oxygen into the cytoplasm of the cell. Now we are going to take the end product of glycolysis, which is the pyruvate molecule, and we will break it down to get more energy. So how that is going to happen? Pyruvic acid needs to leave the cytoplasm of the cell and goes on a cellular organelle named mitochondria, aka ATP factories. So what is mitochondria? What is the structure of this new organelle, and what's the relationship between this new organelle and the breaking down of pyruvic acid to get energy? So as you can see over here, we have the ultrastructure of mitochondria. So as you can see over here in this picture, we have mitochondria. In the picture they are very big, but in reality they are very small, and we can find them into the cytoplasm of the cell. So this is an outer view of this new organelle, but we want to see inside of it. Maybe we can find something that's related to making ATP. So in the second picture over here we have a longitudinal section of the mitochondria so that we can see the inside of it. So to get more information about the composition of mitochondria, let's go to this diagram of the longitudinal section. So mitochondria is composed of two membranes. There is the first one that you can see over here. We call it the outer membrane. So it's a membrane that we can find in the outside of the mitochondria. The second membrane is this one over here that we are going to call the inner membrane. The third component of mitochondria is the space between the outer membrane and the inner membrane that we are going to call the intermembrane space. So for number 5 over here we have what we call the cresta. Cresta are only folds that we can find in the inner membrane of the mitochondria. So these folds are going to increase the surface of the inner membrane of the mitochondria. Number 4, it's the space inside of the mitochondria that we are going to call the matrix. And actually this is where the Krebs cycle reactions are going to happen. Last but not least, these little spots that we can find in the matrix, they are the genetic information of the mitochondria that we are going to call DNA, or the mitochondrial DNA. So the interesting part in mitochondria are these membrane, and especially the inner membrane, because it's much bigger than the outer membrane, and it's giving the mitochondria a really unique structure. So we have over here a section of the two membranes to discover more about them. Over here we have the outer membrane, and this is the inner membrane with the cresta. Let's start first with the outer membrane. The outer membrane is composed of lipids and also of proteins. What makes the outer membrane of the mitochondria is so similar to the membrane of the cell. If you remember, the cytoplasm of the cell is composed also of lipids and proteins. As you know, the famous lipids that we can find in the outer membrane of the mitochondria and also in the membrane of the cell are the famous phospholipids. Phospholipids are broken down into two parts, the head, which is hydrophil, and the tail, which is hydrophobic. In addition to the lipids, we can find the proteins, and especially the transmembrane protein. Now, let's go to the inner membrane of the mitochondria and let's discover the chemical composition of it. So, over here, we can also find phospholipids and we can find also the proteins. In addition, we have here these special structures that are called the ATP synthetase. So what is this ATP synthetase and why we can find it only in the inner membrane of the mitochondria, not in the outer membrane? So ATP synthetase, it's an enzyme. Anything that ends with ase, it's enzyme. It's a protein. For the structure of it, we are not going to get into a lot of details, but only we have two main parts. The first one over here is what we are going to call the router, and the second part is this part that is binding to the inner membrane of the mitochondria that we are going to call the root. The main thing I want you to remember is always this part, the router of the ATP synthase, looks into the matrix. Just put in mind this information. Concerning the function of ATP synthase, it's an enzyme that produces ATP. So after this introduction, we are going to go now into the reactions of the Krebs cycle. Well, as I said in the beginning, pyruvate or pyruvic acid is going to leave the cytoplasm of the cell and get into the matrix of the mitochondria to be broken down into energy. Well, before getting into the Krebs cycle, we have a preparation step where pyruvate, as you can see over here, that we are only presenting with the 3 carbon atoms, is going to bind to this new molecule over here that is coenzyme A. The association between these two molecules is going to produce a new molecule with only 2 carbon atoms, which is the acetyl coenzyme A. So as you can see over here, we went from a 3 carbon molecule into 2 carbon molecule. This is what we call decarboxylation. So carbon dioxide is going to be produced. And whenever we have decarboxylation, we have also the reduction of NAD plus into NADH plus. Now, after the making of acetyl coenzyme A, we are going to start the Krebs cycle reactions. Well, this 2 carbon molecule is going to combine with a 4 carbon molecule named oxaloacetate. So they fuse together and make a 6 carbon molecule. This new molecule over here is the citrate. The interesting part that happened over here is the coenzyme A that is binding to this molecule will be detached from the acetyl coenzyme A and search for another pyruvate molecule to combine with it and make another acetyl coenzyme A, and the same thing will be repeated. Now, for the next reaction in the Krebs cycle, this 6 carbon molecule is going to be converted into 1, 2, 3, 4, 5 into a 5 carbon molecule. So if we compare the two molecules, we already lost one carbon atom. So this is what we already called decarboxylation. So carbon dioxide is produced. And whenever there is decarboxylation, we have also the reduction of NAD plus into NADH plus. Next, this 5 carbon molecule is going to be also converted into this new molecule that has 4 carbon atoms. So we also have carbon dioxide production and the reduction of NAD plus into NADH plus. Now this 4 carbon molecule is going to be converted into another 4 carbon molecule. These are not the same molecule, they are so different. The only common thing between them is the number of the carbon atoms. And what makes this reaction special is this reaction over here, which is GDP plus inorganic phosphate is going to produce GTP. So what is JTP? JTP is guanosine 3 phosphate. So what is JTP? JTP is an energetic molecule that will bind to an ADP to make ATP. So JTP refers to guanosine 3 phosphate. Now the next reaction is this 4 carbon molecule is also going to be converted to another different 4 carbon molecule, but in this case FAD will be reduced into FADH2. And last but not least, this 4 carbon molecule is going to be converted to the first molecule that started the Krebs cycle reaction, which is the oxaloacetate. And this reaction accompanies the reduction of NAD plus into NADH plus. And with the making of oxaloacetate, it's going to wait for another acetyl coenzyme to show up to start another Krebs cycle, and the cycle will repeat again. Now what you should put in mind is for every glucose molecule all these reactions happen twice, because in glycolysis we made 2 pyruvic acid. So all what we talked about right now happened for only 1 molecule of pyruvate. So for each glucose molecule, there is 2 Krebs cycles that are going to happen. Now, after all this, if you realize something, in these reactions, pyruvate is totally broken down. So we got all the energy still in it. Now, let's try to count all the sort of energy that we got from the breaking down of 1 molecule of pyruvate. Well, let's start from the beginning. We have over here NADH plus. Also, we have another NADH plus, a third one, and a fourth NADH plus. In addition, we also made 1 FADH2. And let's not forget about the JTP that is going to be converted also into ATP. Now, after the total breaking down of the glucose, we made only few molecules of ATP since the glycolysis reaction. So how can we get more energy? And also we got so many electron transport molecule. So what can we get from all of them? Well, in the next video we will be answering all these questions. So now let's summarize all what you should put in mind for the Krebs cycle reaction. So the first thing you should put in mind is the location of these reactions, and they happen into the matrix of the mitochondria. The second thing is the output of the Krebs cycle, what we got from the Krebs cycle. So as we saw, we have the preparation step that is from pyruvate to acetyl coenzyme A, so we made only one NADH8+. And since we produced in the glycolysis 2 pyruvate molecule, the Krebs cycle is going to happen twice, so we are going to be producing 2 NADH8+. The next step is actually the Krebs cycle reaction. So we already made 3 NADH8+, 1 FADH2 and 1 JTP, or we can say 1 ATP. And the same thing, this reaction happens twice. So actually from one glucose molecule we got 6 NADH8+, 2 FADH2 and 2 JTP. Next, the global reaction of the Krebs cycle is the following. So CH3, CO, COOH, which is the chemical formula of pyruvate, plus 4NAD+, plus FAD, plus GGP, plus inorganic phosphate are going to be converted into 4NADHH+, 1 FADH2 and 1 JTP, and also we will have the releasing of 3 carbon dioxides. So this global reaction over here is only for 1 pyruvic acid. So if you are asked to give the global reaction of the Krebs cycle for 1 glucose molecule, you should multiply per 2. Well, before ending this video, the last thing I want to mention is for the Krebs cycle reactions, they only occur in the presence of oxygen. So the pyruvic acid will not get into the mitochondria if there is no oxygen in this cell. So put that in mind. Well, as I said before, in the next video we are going to see how can we get more energy. So see you in the next video with the third phase of cellular respiration.